The adult brain accumulates roughly 1 milligram of iron per year through middle age. This iron concentrates heavily in the hippocampus and substantia nigra, regions where iron-driven chemistry closely tracks the rate of cognitive decline. For decades, the logical response was to strip this iron out of the brain entirely. In 2024, the DIVINE Phase 2 trial tested this pan-depletion strategy using deferoprone, an orally bioavailable chelator. This data visualization shows success. Deferoprone patients saw a 25% drop in hippocampal iron versus placebo. But clinical outcomes contradicted the biomarker. As iron dropped, the right trend line shows ADAS-Cog scores worsening. The drug accelerated cognitive decline. The DIVINE trial exposed a mechanical flaw in the pharmacology of pan-depletion. Because iron is an essential electron carrier required for basic cellular survival, stripping it indiscriminately from the brain induces functional failure. Solving this paradox requires a focus on the specific cellular compartment iron occupies and the precise cell death program it catalyzes, ferroptosis. Formally defined in 2012, ferroptosis is an iron-catalyzed, lipid peroxidation-driven, and regulated form of non-apoptotic cell death. This process targets a specific structural substrate, polyunsaturated fatty acid esters, or PUFAs, embedded directly within the cellular membrane's phospholipid bilayer. As upstream bioenergetic membranes falter with age, the resulting oxidative stress funnels into this specific pathway. Ferroptosis operates as the terminal execution mechanism for the aging neuron. Intercepting this execution pathway to achieve disease modification requires mapping exactly where the brain's iron supply turns toxic. Iron's chemical properties make it both a lethal toxin and an essential requirement for cellular life. To manage this toxicity, cells maintain strict boundaries. This 3D model of a neuronal environment separates iron into distinct functional compartments. We have safely bound iron pools, storage within ferritin cages, and iron-sulfur clusters required for mitochondrial complexes 1 through 4 and dopamine synthesis. Outside these bound systems exists the labile iron pool, a volatile, loosely bound fraction of ferrous ions floating freely in the cytosol. Research at the Florey Institute identified exactly why deferoprone failed. The chelator did not distinguish between these pools. It indiscriminately stripped iron from the toxic labile pool, but also tore it away from the mitochondrial complexes, inducing immediate bioenergetic anemia. The therapeutic goal is selective compartmentalization, neutralizing the labile iron pool while leaving the structurally bound pools required for cellular respiration intact. If the labile iron pool is left unchecked, the pathology it triggers plays out directly on the cellular membrane. The destruction runs through the Fenton reaction. This chemical equation shows labile ferrous iron reacting with hydrogen peroxide to generate a highly reactive hydroxyl radical. Hydroxyl radicals are the most physiologically reactive species in the body. They immediately attack the densely packed PUFA tails comprising the membrane, stealing hydrogen atoms and generating unstable lipid radicals. This initiates non-enzymatic lipid peroxidation. One lipid radical attacks the next, creating a chain reaction that rapidly propagates across the bilayer. Structural lipids break down into chemically unstable lipid hydroperoxides. This cascading structural disruption of the membrane is the proximate driver of ferroptotic death. Neutralizing it requires a specialized defense system integrated directly into the membrane structure. The cell's primary defense against this structural collapse is the enzyme glutathione peroxidase 4, or GPX4. This diagram shows GPX4's unique mechanical capability. Unlike classical antioxidants that float in the cytosol, GPX4 is the only enzyme that can physically reach into the membrane bilayer, extract a lipid hydroperoxide, and reduce it to a harmless alcohol. GPX4 relies on strict upstream dependencies to maintain its catalytic rate. Its active site is built from a selenocysteine residue, requiring continuous dietary selenium, and the reduction process itself rapidly consumes the cellular reductant, glutathione, or GSH. If the GPX4 axis is overwhelmed, the cell relies on a parallel defense. This diagram shows the membrane-associated FSP1 protein reducing CoQ10 into ubiquinol. Ubiquinol acts directly inside the membrane as a lipid-soluble radical trap, neutralizing peroxyl radicals independently of GPX4. Stabilizing these lipid hydroperoxidases is mandatory. When iron chelation fails to capture the full Fenton flux, GPX4 and SFP1 stand as the final barrier preventing membrane collapse. Ferroptosis is a systems-level failure where diverse upstream bioenergetic deficits mathematically converge. This systems biology flowchart maps the forward coupling between membrane failure and energy failure. Unchecked lipid peroxidation produces 4-HNE, a reactive electrophile that migrates to the mitochondria to permanently adduct and destroy the catalytic core of ATP synthase. The coupling runs in reverse as well. As shown on this descending branch, upstream NAD+ depletion breaks the pentose phosphate pathway, halting the cellular supply of NADPH. Without NADPH, the cell cannot regenerate its GSH supply. This starves the GPX4 enzyme, leading to deactivated defenses and runaway lipid peroxidation. This feedback loop creates a terminal systemic collapse. Energy failure accelerates membrane destruction, and membrane destruction permanently poisons energy generation. Because this bidirectional spiral connects mitochondrial function directly to membrane integrity, effective disease modification requires a multi-target regimen. The matrix maps a combinatory strategy. Quadrant 1 targets selective label iron chelators that reduce Fenton flux without depleting mitochondrial iron. Quadrant 2 addresses defense, stabilizing GPX4 through targeted selenium supplementation. Quadrant 3 provides parallel redundancy, activating the FSP1 pathway and supplementing CoQ10. Modern amyloid clearance therapies dissolve plaques that historically functioned as iron sinks, which can paradoxically dump sequestered iron straight back into the label pool. Intercepting the ferropodic cascade requires these interventions to compose multiplicatively, attacking the process from both upstream and downstream simultaneously. The diagnostic tools to execute this combinatory strategy exist today on the immediate clinical horizon. Quadrant 4 maps patient stratification. Measuring routine plasma selenium identifies patients below the 100 microgram per liter saturation point, the demographic requiring GPX4 support. Genetically, ApoE4 homozygotes carry elevated membrane PUFAs and higher iron, while PSEN1 carriers exhibit impaired selenium uptake. A combinatory clinical trial co-administering selective chelation, GPX4 stabilization, and CoQ10 in these stratified cohorts will produce cognitive benefit that exceeds any single molecule intervention. By addressing the pharmacology of specific cellular compartments, we shift from passively managing upstream proteinopathy to directly intercepting the terminal executioner of cognitive collapse.